given incredible talks already this morning, so very high bar. So my background, I'm a neurosurgeon. I treat patients with spinal cord injury. And the frustration is that there's not much I can do apart from rods and screws to put them back into place. But their nerves don't grow back. And that sort of set me on a path of explorer and science. I started as a stem cell biologist and it turned to a synthetic biologist. On that path, I found out that there's a crisis that affects the development of new drugs. At this point in time, we spent about 2 billion to create a new drug. Our knowledge in science and biology is increasing exponentially, but the failure rates have hit a near all-time high. So how do we tackle that? There's some good news. We are also seeing a transition from traditional medicines based on small molecules and biologics to new generations of medicine, such as the base editing that we've just heard of, and the concept of using cells as medicines themselves. This is called cell therapies. And the amazing thing about cells is, of course, that they're intelligent. They can replace lost cells, and they can also interact with their environment. So the first cell therapy that's now available is able to cure blood cancers, which were untreatable before. And last year, the first patient was cured of diabetes with a transplant of pancreatic islet cells. But here's the challenge. At the moment, these therapies cost enormously high. So 500,000 for a treatment of a blood cancer. I think the NHS pays £375,000. And the manufacturing is a nightmare. And these therapies aren't scalable. So how do we solve that problem? And we need to rethink biology. This is Sid Brenner. He won the Nobel Prize for his programmed cell death discovery. He wrote in an article, we need to move away from the traditional questions of biology. How does it work? How is the cell composed? And how did it get there? To a new paradigm in biology in which we realize that the code script is the center of biological theory. In this paradigm, we can look at the cell like a piece of hardware. In the middle is the hard drive. It contains the DNA, and the DNA contains the genes. These genes interact with each other to form gene regulatory networks, the genetic programs that instruct the cell and instruct the function of the cell. And this code, as you know, is translated into the physical component of cells, the proteins, the lipids. So this is a beautiful depiction of the inside of a cell. At the top, you see the DNA, then there's a membrane, nuclear pores. On the left, you see a mitochondrion. This is the energy hub of a cell. And the thing to note is the software that is encoded in the DNA translates into the physical reality. If you change a gene, if you turn on a gene, the cell, the physical reality of the cell will change. So this is what I mean by programming biology. And that's not entirely new. In fact, we started interfering in programming biology about 10,000 years ago when we started to select plants and animals, domesticate them. In the 1700s, we learned how to hybridize plants. And nowadays, nearly everything that we eat has been modified by us human beings. In the 1800s, we heard about genetic inheritance and, of course, evolution. In the 1950s, we discovered the structure of the DNA. And then shortly after that, in the 1970s, we found the first restriction enzyme. This is a very simple way of cutting the DNA but it started the biotech revolution that we've seen over the last 40 years and enabled, for example, the manufacture of insulin as a drug. In the 1980s, we started to apply our tools to the modification of plants. 1990, who remembers Dolly the sheep? In the 2000s, really, that's when another transition started. We started to read DNA. We sequenced the human genome. And then 2010 and plus, we found tools like CRISPR and base editors to really sort of write DNA. Around the same time, we also learned how to actually execute genetic information in cells. So reading, writing, and executing code, this is really the paradigm of synthetic biology, of programming biology. And we've used this to create new classes of drugs. We talked about biologics, where we had to engineer lower species such as E. coli and yeast. And these are now programmed to deliver these modern medicines. But there's always been a barrier, the barrier of applying the same tool of technologies that we have developed to higher species. Nevertheless, we are on a critical point in time where biology is undergoing a transition that we've perhaps seen before in a different context. So if you look back at physics, 500 years ago, it was a descriptive science. People were observing the objects of physics. But then everything changed when we started to introduce maths. Because maths gives you a lens into the future. As you can see, Leibniz and Newton, and of course, the introduction of calculus. And that sparked waves of innovation from steam engines to rockets to spaceships. But we've then sort of undergone a second cycle in which we transitioned from physics to computer science. And you can see Ada Lovelace here, who invented the first code. And of course, we all know over the last 40 years how this has transformed our economies. So biology is at the cusp of transitioning in a very similar way. And the gate of opening up programmable biology of human cells is really one of the technologies that are the foundation of my company, BitBio. We're using what's called genetic safe harbors. These are specific sites in the genome in which we insert genetic information. And the interesting thing about these genetic safe harbors is that they keep the cell safe. So it doesn't interfere with the genetic program, but they also keep the program safe. So you can activate it like a light switch. And what this allows you to do is you can program stem cells, for example, into, in this case, neurons, so brain cells. And this opens up the industrial production of mammalian cells, including human cells. So this is how it looks in real life. You can start with pluripotent stem cells, and we execute a program that defines a neuron, which is a brain cell that connects with other brain cells to exchange electric information. And you can see within 4 days, they reach that stage, they start to connect to each other, and they form a real neural network, none of the sort of artificial ones in our computers. And not only that, they mature to an extent that we've not seen in stem cell biology before. Now, you think that's probably a one-off. That's what I thought when I first saw what my students have done in the lab. But actually, since then, we've been able to apply it across different cell types. Important for scientists, 3 germ layers, the 3 origins of all the tissues in our body can be programmed. And you can see a variety of cells that match that. Now, if you think about it, disease always strikes at a cellular level. And in turn, every cell is a potential medicine. So that's the possibility that we're seeing in front of us here. So there are other use cases for human cells, of course, as well. One is the problem with drug discovery. At this point in time, we are using non-human cells, animal cells, and cell lines to create drugs for Alzheimer's. And what we learn over and over again, that the biology is fundamentally different. Imagine you can use now highly consistent human brain cells that are actually affected by the condition. This will increase the odds and therefore lower the price for new drugs. As I said, I'm super excited about the prospect of using cells as therapies. And up here, you can see Emily, the first child that was treated with a CAR T cell therapy. I would call this is the Brother Wright flight of cell therapy. Very simple modification done in a paper, like a sticker plus way. And nevertheless, she's been cured. 9 years later, she's free of cancer. You can go beyond cell therapies and think about combining cells to organ systems or linking them up with electronics to really form bionic devices that can be hooked up to the body and controlled by our brains. And as you said before, we can also leave this incredibly exciting future of medicine to look into other areas where this might play a role. Let's take, for example, cultured meat. The ability to grow meat at scale in a lab will address a number of problems that we're facing at the moment. The environment, methane, and of course, the use of antibiotics in high-density farming areas. So I do think that this transition of biology into a programmable biology will really open up an incredible amount of opportunities and transform our world.